physicist Niels Bohr once said that prediction is really hard, especially about the future. And with that in mind, I am going to avoid my initial thought of predicting where we're going to be in 25 years. That is just not possible. But instead, what I decided I would do is talk about where we are at this moment and where I see us headed over the next 5 or 10 years as an idea of the direction of where we are going. And I think that is maybe feasible. And so we'll start at the beginning, when the founding of the Institute 25 years ago by Pat and Lori McGovern and under the founding direction of Phil Sharp. And I think it would be fair to say that none of the people involved could have imagined where we would be today. And if you reflect back on where we were in those beginning days, and that's a picture, that's not the very beginning faculty, but an early picture of the faculty. It includes Chuck Vest, the president of MIT at the time in the picture. I would say, and this was started by Phil really, when he assembled those core faculty members, we had a molecules to mind approach, which was you go from the basic building blocks, and we had Bob Horvitz studying molecular genetics. And then you go through all the intermediate steps. And for example, we had Jim DiCarlo recording from individual neurons using technology that really had been invented by Hubel and Wiesel and the cat. And we had, in the cognition, and we had Nancy Kamwisher studying perception and cognition. And that's where we were then. And if you think where we are now, well, first of all, we've greatly expanded the faculty, many new members. And I have to say that besides their brilliance, I think one of the key defining features of the faculty is they're just really nice people, and we just like working with each other. And then you think about all the steps now, it's like there's just an explosion of information that we have about all these different steps. So if you think about the molecular side, we now have this a tremendous understanding of genes that are important for both health and disease. And who could have imagined back then that by now we would actually have methods for editing the genome and correcting mutations that are found in some diseases. And this is an area this is not like it happened and then we moved on. This is an area where Feng Zhang continues to develop new genome editing technologies practically every month. There's some new discovery coming out of his lab, and this is only going to continue into the future. And so now we have this tremendous genetic understanding and this ability to manipulate genes. And now, moving on from that, we're now beginning to understand gene expression across the brain and in different cell types, in fact, defining many of the different cell types that we have in the brain. And we're beginning to get an understanding of how this gene expression changes in disease. And that's only going to greatly expand in the upcoming years. But gene expression leads to proteins, of course. And so now finally we get to visualize it all. We have expansion microscopy from Ed Boyden, which first allows us to view cells and the interior of cells in unprecedented detail. But very recently, he's developed the methods for imaging proteins in those cells. And in just the next few years, I think probably sooner than that, but Ed will have ways of visualizing proteins in living animals in vivo, in animals learning and memorizing. And so that we will begin to be able to see the changes taking place within cells that allow us to store memories and learn new information. Now, if we go one step further to the recordings, as I mentioned in the earlier slide, when we started off, people recorded from one neuron in the brain at the time, and that's because that's the technology that we have. But now we know that an individual cell contributes almost nothing to any thought or behavior, that they're working together with millions of other cells in networks that are distributed across the brain. And so if we really want to have any kind of deep understanding of thoughts and behaviors, we have to get this bigger picture of what all these cells are doing that are producing this. And so now we have technology, for example, like in neuropixels, and Mirdad Jazieri is spearheading the use of neuropixels here, that can allow us, right today, hundreds of neurons at a time, but certainly in the next few years, it'll be thousands of neurons at a time, and distributed across multiple brain structures to give us this really complete picture. And even now with the technology being developed by Polina and Akiva, we have the ability to record from neurons outside the brain in previously inaccessible structures, like even the gut. And so the neuroscience of the future is going to be based on this much more complete understanding of how neurons are contributing behavior throughout the entire nervous system. Now, when I joined the McGovern Institute 20 years ago, optogenetics was not a thing. It hadn't been invented yet. And since then, of course, we've had the invention of optogenetics by Ed Boyden and his collaborators. And it's now grown so that you can hardly run an animal neuroscience lab now without the use of optogenetics for testing causal hypotheses about how all these neural circuits are working. It's one thing to see correlations between behavior of neural circuits and behavior, but we're often fooled by correlations, and so you want to be able to get in there and test them by manipulating those circuits, and optogenetics gives us this tool, and we're even now, and this is only going to increase in the future, beginning to see clinical uses of optogenetics, so for example, in potentially curing blindness. Now, so I think you're probably convinced by now we have this explosion of data, but how do we make sense of it? And it reminds me of the dilemma that was faced by the early astronomers, who had tons of observations on the movement of the planets through the skies, but they didn't understand why they moved that way until we had the models of Kepler and Newton that had developed the models and theories of why planets move the way they did. And then it all began to make sense, and that you can make accurate predictions and so on. And that's where we need to be in neuroscience. We need those models and theories to understand how all these neural circuits work. Now, this is an effort that was pioneered by Tommy Poggio, and now we have examples, for example, from Ila Fiet, who has the model of navigation based on the properties of cells in the entorhinal cortex. We have the models of bird learning from Michael Fee. We have models of perception from Jim DiCarlo and so on. And now from all these fellows working in the Yang Tan Icon Center, we have people working on these neural circuits all over the brain. And I think what we're going to be seeing over the next few years as we build up our understanding of all these different circuits, I think we're going to have at some point in the not too distant future an idea of how the brain works as a whole, a theory of brain circuit function. And that now leads us to, where are we in human neuroscience? Now this I want to spend a moment on. When the McGovern Institute started, we didn't even have a brain imaging center. In fact, when I arrived here 20 years ago, we still didn't have a brain imaging center. Nancy Kamwisher was trekking over to MGH and standing in line trying to get her subjects imaged over there. But Phil Sharp had wisely planned for an imaging center in this building. And then finally it got going. And since then, over these years, we've had an explosion of studies of human perception and cognition using functional brain imaging, which it includes some functions that are really specific to human beings and would be very difficult to study in animals. And that includes language, for example, being studied by Ev Fedorenko, theory of mind being studied by Rebecca Sachs, and many other different aspects of perception and cognition and so on, and even some aspects of brain disorders, including autism and schizophrenia and depression, and from the work of John Gabrielli, who's trying to find brain imaging markers that could predict who would benefit from certain types of treatments, something that we call precision medicine, customizing the treatment to the individual. And this is something, this precision medicine effort is something that I think is going to be increasingly important in the upcoming years. Now, where's human neuroscience going in the future? And I see that human neuroscience is going to benefit from technologies that are beyond fMRI and using some of the technologies that are more closely related to what we've traditionally been able to use in animals. And the one example of this is from the work of Mark Harnett, who based on collaborations with neurosurgeons at MGH, he's able to rush over to MGH if they have a neurosurgical patient where they have to remove some brain tissue. And if the patient volunteers, Mark can take that brain tissue, grab an Uber, rush over here to MIT, and bring it to his lab and study these neurons in slices. And he's now beginning to identify what are the features of these human neurons that are important for us as humans, as human beings, and that differ from some other species. And not only are we able to do this sort of biophysical work in humans, but we'll also now be able to record from neurons in the living subjects. So these are in subjects that they're epilepsy patients, and the neurosurgeons have implanted electrodes so you can record neural activity. The neurosurgeons are trying to use these electrodes to find the seizure focus, but if the subject is willing to participate in experiments, we can do these neural recordings. So for example, with Ev Fedorenko and Nancy Kanwisher, we can record from neurons in the face area, for example, and see how they process information. We can record from neurons in the language area and see how they respond to sequences of words and process information. And in a new direction, I'm very excited about myself, is that we now have the ability to study the anatomical connections of these neurons in a way we never had before, using some techniques that were developed in animals where you can combine electrical stimulation with fMRI. You can actually see real anatomical connectivity in these studies. And so now, for example, if we're looking at the face area, we can do the stimulation with recording and see what is the information coming into the face area and where does it go? And likewise for language, what is the information that goes into these language processing networks and where does that go? And so these are early days, but I think what you're going to be able to see in the upcoming years is this beginning of a kind of neuroscience in humans that we only used to be able to see in animals before. Now, another major advance that's affecting human neuroscience, of course, is the development of artificial intelligence systems. And there's now a great interest in comparing and contrasting how these artificial intelligence systems work compared to our actual biological brains. And one of the many things that people here are looking at is the modular architecture of some of these artificial systems compared to the modular architecture of the brain. Nancy Kanwisher, Josh McDermott, Ev Fedorenko, all looking at the development of this modular architecture in the artificial and biological systems. And Jim DiCarlo and Nancy Kanwisher even have a paper in which they show that under some conditions, an artificial system can even develop the face patches like we have in the human visual system. Now, just imagine that, let's say over the next 10 years, that we have what's called artificial general intelligence, where we have artificial systems that actually equal or even surpass human intelligence. I think the insights we're going to get by understanding these systems are going to give us real insights into understanding the human brain as well, and both in terms of similarities and differences. Now, where else is artificial intelligence being used? Well, certainly in therapeutics. So now we can take the information we have about the genetic and molecular architecture of disease, and artificial systems are already in daily use in our labs. That are tools like AlphaFold, for example, predicting proteins. And these tools are going to accumulate to help us bridge between the architecture of disease and then the therapeutics that can target those disease structures and actually have a beneficial effect. And this is something that's going to be much more common over the next few years. And this is in the context of a broader program that we have in therapeutics. We have a whole group of faculty members working in addiction, for example, led by Fan Wang and including Anne Graybiel and Ellen Jasanoff, John Gabrielli and Polina Anakiva, all studying mechanisms of addiction with the goal of therapeutics. We have Fan Wang's work in understanding pain with the goal towards therapeutics. We have Yu He working to develop prosthetic limbs, and Nidhi Sithapathy working on modeling human body motion so that potentially humans could have robotic appendages in the future. And then, of course, all the work going on in autism, schizophrenia, depression, anxiety, of course, work that's really in the biology end, spearheaded by Guoping Feng. But in humans, we have, as I mentioned before, John Gabrielli looking for ways of predicting treatments that would be effective in specific individuals from people suffering from these disorders. Now, so if you look at what are some of the signature accomplishments of the efforts in therapeutics, of course, there was the FDA approval of Casgevy as a gene therapy for sickle cell disease. And now we have this clinical trial that was approved using Guoping Feng Chang3 mini gene in human patients for treatments in Phelan-McDermid syndrome. And the first patient received their gene therapy just last month. And what you're going this is just the beginning. We know not just here, but outside of MIT, there are many trials underway using CRISPR-based technologies, and I think what we're going to be seeing in the next era is the era of gene therapy that's going to include the brain, and not only will there be the CRISPR-based gene therapies, but there's going to be a whole variety of therapies in our gene therapy toolbox that will be needed to address different kinds of genetic abnormalities. Those include gene silencing and modulation, the genome editing, of course, and gene replacement and addiction. Now, I have to qualify the gene therapy efforts here by saying that these are all directed at what we call monogenic disorders. These are disorders that have individual genes with mutations of very big and usually bad effects. And so they provide a clear target for gene therapies. But these are only a small part of basically human medical conditions. And a lot of human diseases are not due to don't have a monogenic cause, but have what we call polygenic causes. Dozens or hundreds of genes all contributing to the risk for developing these disorders. So how are we going to help them? Well, one way, following this approach, is that I think that it will have in the future is as we gain a better understanding of the neural circuit abnormalities in these monogenic disorders, our hope and belief is that some of these circuit abnormalities will be shared with the polygenic forms. And then taking what I've told you about our greatly increased understanding of how neural circuits work, we're going to be able to modulate these circuits to produce benefits in people even suffering from polygenic disorders. And there are many disorders have this polygenic basis, and just autism, schizophrenia, and depression are just examples of these many disorders that are polygenic. Now, another therapeutic direction I want to mention that I happen to be personally interested in is our ability to help aging. Just one example is Mark Harnett has identified these tiny synapses, the filopodia. They're sometimes called silent synapses, which are recruited into forming new memories and learning. And Mark has found that these, surprisingly, these persist through our adulthood and seem to be important for our forming new memories and learning through the lifespan. But, and here's the bad news, is we lose them as we get older. And so obviously this presents a potential therapeutic target for how can we prevent the loss of our poor philopodia and preserve some of our abilities to learn and remember as we age. And an even broader approach is being taken by Feng Zhang, all new approach in his lab to addressing aging. And what Feng is trying to do is to rejuvenate different organs and systems in the body. And he's starting with the thymus and the immune system. Now, the thymus is a critical component of the immune system, and we're born with a big, healthy thymus. But as we age, that poor thymus shrinks. And so the hope is that with this rejuvenation, he can restore some of the functions of the thymus and rejuvenate the immune system, which will have beneficial effects on many organs of the bodies, including our brains. So I hope that with this very, very brief view of where we are and where we're going, I hope you'll agree with something that Pat McGovern used to always say about the McGovern Institute, which is the best is yet to come. And if we're successful in the Anti-Aging Institute, I hope to see you back here in 25 years. See you in 2050.